honorable judges, I'm Feng Jingqiu from Chinese Naval Medical University. It's an honor to present our research topic here, study on the mechanism of BRCA1 involvement in placental trophoblast cell differentiation. Today, my presentation includes 4 parts as you can see on the screen. Now, let's begin with the background. As we all know, placenta is a vital organ during pregnancy, ensuring the normal development of the embryo. Most cells in placenta are trophoblasts. The trophoblasts include EVT and HCT, both differentiated from CTB. Among them, CTB has stimulus, and EVT has high invasiveness and migration. So, abnormal differentiation of trophoblast stem cells into EVT can lead to various pregnancy complications. So, EVT is the focus of our research. In addition, the transcription factor, BRCA1, plays a crucial role in the differentiation of various tissues. However, its role in trophoblast differentiation remains unclear. So, our research aimed to explore the function and the mechanism of BRCA1 in trophoblast differentiation. For this purpose, we have conducted a series of experiments. Our technical approaches are as follows. Firstly, we conducted clinical sample analysis to find key genes and pathways. Secondly, we conducted in vivo and in vitro experiments to validate the effect of BRCA1. Finally, in mechanism studies, we conducted RNA sequencing to investigate the mechanism of BRCA1 and validate it through WB, qPCR, and rescue experiments. In the early stage, we analyzed the single-cell RNA sequencing data of placentas from normal individuals. We found genes related to trophoblast differentiation are enriched in this pathway. Among them, BRCA1 is specifically expressed in the initial stage. Especially, BRCA1 is highly expressed in the early stage in all samples, gradually decreasing with differentiation, and BRCA1 is particularly expressed in the progenitor cells and early proliferative EVT. BRCA1's target genes are enriched in many pathways. However, the Wnt pathway is associated with the maintenance of stimulus and cell differentiation of trophoblasts. And these BRCA1's target genes are enriched in Wnt pathway. So, we can infer that the BRCA1 Wnt pathway may be the key pathway. To verify the function of BRCA1, in vivo, using conditional gene knockout system, we knocked out BRCA1 in trophoblast stem cells. This resulted in increased junction neuron cells, decreased labyrinthine neuron cells, and abnormal enlarged vascular lumen. Among them, J cells has high invasiveness and migration. In vitro, shRNA-mediated lentiviral interference was used to silence BRCA1 in HTR8. Cytological experiments indicated that silencing the BRCA1 enhanced tubulogenesis, proliferation, and apoptosis, coupled with reduced migration and invasion. So, based on the results of in vivo and cytological experiments, we can indicate that BRCA1 knockout may cause excessive proliferation and reduce differentiation of trophoblast stem cells, hindering their differentiation into mature invasive trophoblasts. And this can cause shallow placental implantation and various pregnancy complications. To verify the mechanism of BRCA1, we analyzed the RNA sequencing data of HTR8 and validated them through WB and qPCR. The results showed that a significant downregulation of Wnt pathway after BRCA knockout. In clinical sample analysis, we found many BRCA1's target genes are enriched in Wnt pathway. However, only FCD6 showed a significant decrease after BRCA knockout in both RNA and protein levels. In addition, many studies have reported that BRCA1 is a receptor on the Wnt pathway. So, we detected the changes in key proteins of Wnt pathway, and the results also showed a significant decrease. So, we can infer that BRCA1 may regulate the Wnt pathway through FCD6. To validate the FCD6 is indeed a target gene of BRCA1, we conducted a cheap qPCR, and we identified the regulatory site of BRCA1 on FCD6. To investigate which functions of trophoblasts are affected, we designed a rescue experiment. Firstly, we silenced BRCA1 in HTR8, then overexpressed FCD sacs, and finally conducted cytological experiments. The results show that overexpression of FCD sacs could reverse invasion and migration, but could not reverse other ability of cells. So we can infer that BRCA1 FCD sacs wing pathway may affect the invasion and migration of trophoblasts. All in all, we can control the following conclusion. BRCA1 is one of the key regulatory factors affecting the differentiation of trophoblast stem cells into invasive trophoblasts. And BRCA1 may regulate the wing pathway through FCD sacs, affecting the differentiation of invasive trophoblasts. So, any abnormalities in this process can lead to disorder placental implantation and various pregnancy complications. Of course, our current research mainly focuses on EVT, but in the future, we will continue to focus on syncytial trophoblast and trophoblast blood vessels to improve the regulation and provide treatment basis. That's all for my presentation. Thanks for your listening.
Wow, such an interesting topic. Do you have any comments or any questions?
Sorry, can you elaborate more on how you select the samples for the single-cell sequencing experiment?
Okay. Here, we analyzed the single-cell RNA sequencing data of placentas from the normal individuals. However, our current research do not focuses on the placentas from individuals that has pregnancy complications. But in the future, we will continue to validate the effect of BRCA1 to analyze the single-cell sequencing data of people that has pregnancy complications. And we found BRCA1 is highly expressed in the early stage. That means BRCA1 expressed differently at different ages of trophoblast differentiation. But other genes do not have these characteristics, so we think BRCA1 is the key gene.